Hi everyone, I'm Colleen.
And I'm Jasmine.
And today we'll talk about detecting the immune system response of a 500-year-old Inca mummy.
A mummy is a human or animal that has been preserved by removal of internal organs.
So, forensic techniques such as DNA extraction and PCR amplification have provided evidence for historical questions such as death of Tutankhamen.
DNA techniques is important in this context because it can determine if the cause of death is deliberate or natural. Thus, it can change how we view a historical event.
Detection of pathogen is needed, but it is not enough to determine the disease since it can be present without causing any infection. However, detecting the immune reaction to the pathogen provides positive evidence of active pathogenic infection.
Existing methods such as antibody-binding immunoassays require fresh samples and uses a small number of targeted antibodies and are inclined to false positives and negatives.
So, proteomics is much better because proteins outlive DNA by thousands to millions of years.
And protein detection does not rely on amplification, so it is less susceptible to contamination.
Lastly, broad spectrum of proteins can be characterized from small samples, resulting in a more resolved picture from immunoassays.
3 mummies were recovered in the summit of Llullaillaco in the province of Salta, Argentina. 2 were young children at the age of 7 and 6, and 1 was a 15-year-old girl known as the Maiden. They were sacrificed to Pachamama, the earth goddess, in the ritual of Capacocha, which means they were killed either by strangulation, heavy blow to the head, or buried alive after being intoxicated. The figure shows the children of Llullaillaco.
So, the bodies were in outstanding condition because of the freezing temperature, mild humidity, an aerobic environment, the presence of natural disinfectants, which inhibited the decomposition of the bodies. Volcanic ash inhibited the growth of decomposing bacteria and fungi and acted as a barrier to moisture. Lastly, a layer of snow made the tombs airtight.
Mouth swabs were taken from the lips of the maiden and both with blood and saliva. A small blood-soaked piece of cloth was taken from the boy's cloth. The girl was not sampled due to signs of being struck with lightning.
The samples obtained were stored in sealed vials to avoid contamination. Samples were kept dry to avoid oxidative and hydrolytic lesions to the DNA.
For the proteomic analysis, these processes were performed.
To distinguish the protein expression profile of an active infectious state from the differential protein degradative state, the boy was used as a control since he showed no signs of respiratory diseases, contrast to that of the maiden.
The proteins recovered were divided into inflammatory and immune response and all other proteins.
Wilcoxon non-Whitney test was used to compare the spectral counts of the maiden and the boy.
To presume respiratory infection with the maiden, her proteomic profile should show significantly higher levels of inflammatory and immune response proteins than those of the boy.
DNA processing was carried out. DNA extraction was done in a sterile environment. PCR was done with 4 sets of primers that targeted 16S rRNA, MTP40, and HSP65 of Mycobacterium species.
No amplifications on the negative controls, which meant that the experiment was good.
Out of the 16 PCR reactions for the maiden, 8 were positive and all the 7 PCR reactions for the boy were negative. Out of the 8 positives in the maiden, 2 were successfully sequenced and showed a Mycobacterium and Bifidobacterium species heat shock protein genes.
These sequences were identified using phylogenetic approach. To determine the probability of misidentifying the sequences, they used Bayesian probability, posterior probability, and weighted Shimodaira-Hasegawa tests.
So for the results, we will briefly discuss the radiological and visual examinations of the maiden and the boy, as well as the results of proteomic analysis taken from the mouth swabs.
The CT scan of the maiden revealed that while all her organs were intact, a number of complications were found to be linked to infectious diseases. The upper lobe of her right lung was radiolucent, a sign of bone resorption. There is also mucosal thickening in the maxillary sinus, which is an evidence of sinusitis. A zoster-like lesion in the right calf was also seen, as well as a mucus streaks under the nostrils.
Upon examination of the boy, there were no signs of upper respiratory tract infections, lesions, and mucosal enlargement. Next, they took and examined the mouth swab samples from both mummies to determine the proteins and check the presence of pathogens. Using a high-resolution mass spectrometer, nasal secretory and blood proteins were revealed in both mummies. In the maiden's mouth swab sample, the nasal mucus protein level were twice as high as that in the boys.
They also discovered several immune response proteins linked to infectious diseases. Cathepsin G, which causes inflammation, is linked to pathogenesis of pulmonary-related diseases. Neutrophil elastase, which function to destroy virulence factors and kill bacteria. Alpha-1 antitrypsin, a sign of mycobacterial infection. Lastly, neutrophil defensin 1 and 3, which are particularly connected to the lung inflammatory response of macrophages.
In addition, proteins involved in severe lung inflammatory response were discovered. S100A8A9 apolipoprotein, A1A2, and transthyretin proteins are associated with chronic and acute lung inflammation. VDV, serpin, and TTR proteins can also be expressed in acute lung inflammation, but particularly in mycobacterial infections.
The presence of this set of proteins, along with the CT scan and visual examination test, supported the idea that the maiden contracted pulmonary infection before her death.
The amount of immune and inflammatory response proteins in the maiden were higher than the boys, where they saw a median spectral count of 44 in the maiden. The boy had none. Lastly, to find out the source of the disease, they performed a PCR assay with mycobacteria-specific primers and extracted DNA from the mouth swab to amplify the HSP65 gene.
Sequencing results show the presence of Mycobacterium species and non-pathogenic Bifidobacterium species. The latter in the sample may have resulted from vomit prior death, rather than a result of post-mortem contamination.
After performing a phylogenetic analysis on the Mycobacterium sequence, they compared the best phylogeny to alternative trees to come up with the best placement of the sequence. By doing so, they ruled out cross-contamination of Mycobacterium species sequence from Bifidobacterium species and other non-pathogenic strains of Mycobacterium. In the end, they determined that the sequence of pathogenic Mycobacterium avium bovis tuberculosis complex had the higher probability to cause the disease.
On the other hand, the blood stains from the mouth swab and cloth of the boy were interpreted as signs of trauma and bleeding. The nasal mucus protein level was found to be normal. He also had high levels of alpha-1 antitrypsin and low levels of neutrophil defensin, which suggests that he did not carry any lung infection. Other than the serum proteins, only the spectral counts for the immune response proteins were significantly different than the maidens.
So, for the discussion, it was concluded that ancient Andean people suffered from mycobacterial infection, but mycobacteria such as Mycobacterium tuberculosis can survive for so long without causing infection. Hence, detecting presence of pathogen doesn't always mean active infection. This study gives direct evidence of active antibacterial immune response at the time of death of the maiden compared to that of the boy.
Radiological examination of the maiden's lungs showed overinflation and trapped air, which are common in patients with mycobacterial infections, particularly Mycobacterium avium and tuberculosis.
This study focused on archaeological specimen, but potential forensic applications include characterizing the physiological state of the source of blood in criminal cases. We can also ascertain if a person is sick or just carrying a latent form of pathogen.
Forensic proteomics offers a sensitive but less contamination-prone alternative to PCR amplification for ancient samples. Immunoassays can detect active immune response and infer infection in historical samples, but it has low specificity and sensitivity, unlike shotgun proteomics, which can play a vital role in pathological determination of the cause of death or disease in archaeological and criminal cases.
Although published 8 years ago, this journal remains significant in terms of detecting an active state of infection prior to death in an ancient sample. This would be helpful in forensic legal studies.
This journal also gives insight to the ability of shotgun proteomics to effectively scope out the pathogens needed for detection in a DNA assay. Additionally, it can serve as a useful tool to assess the physiological state of an organism by looking at the protein abundance information.
focus on specific protein of interest instead of using shotgun proteomics. More exact and sensitive quantitative techniques must be used, such as MRM on a triple quadruple tandem mass analyzer or by comparing the results to stable isotope payload standards.
For further studies, a paper published in July 2020 about non-invasive paleoproteomics of mummified human skin is a good example of how protein sequence is analyzed from mummified individual without influencing the integrity of the body.
In this paper, they used a non-invasive sampling technique based on mixed bed chromatographic media embedded on EVA, which had been used on historical material and was successful in extracting ancient proteins from the surface of Egyptian mummies, and this showed that it is possible to obtain metaproteomes from the surface of mummified body in a simple, rapid, and non-invasive way. Hence, we can target more number of specimens and conduct intensive sampling. And that will be all.
Thank you.